succeeded to arrive approximately on time, but it's quite okay. Directly from Italy. So let me say a few words about her. So, yeah, Professor Chiocchetti holds PhD in biology and medicine, and is a full professor of immunology at the University of Eastern Piedmont in Novara, Italy. Her extensive expertise spans immunology, with a particular focus on the development and preclinical characterization of recombinant proteins and antibodies for immune system regulation, both in vitro and in vivo on animal models, as well as in clinical trials. Additionally, she is highly proficient in high-throughput analysis. Throughout her career, Professor Chiocchetti has supervised 15 PhD students and numerous master theses, demonstrating her dedication to advancing academic research and mentoring the next generation of scientists. Currently, she serves as a coordinator of the Horizon 2020 project, FLAMIN-GO, which is convening its 6-month meeting these days in Ljubljana. This groundbreaking project aims to revolutionize clinical testing approaches for drugs targeting rheumatoid arthritis. So we're delighted that Professor Chiocchetti has joined us today to share her insights on the challenges and prospects of this very exciting topic. So join me in welcoming Annalisa Chiocchetti, and the floor is yours, Annalisa.
Thank you very much. So thank you very much, Sasha, for this invitation, for giving me the opportunity, let's say, to tell you something about our project, but not only. So we chose this topic, which is precision medicine, trying to understand if really it will hold the promises and it will be the solution for many, let's say, unmet clinical need, especially related with the treatment. So I will start my presentation with this picture. I love it a lot because this picture shows the 10 most used drugs in the United States. It's old, it's a bit old, some years ago, but the message is always the same. So in blue, you see they can be for autoimmune disease, heart, pressure, depression. So very widespread treatments, but the message is the same for all. In blue, you see the responder, so people who benefit from the drug. In red, the one who take currently the drug, but they don't benefit at all, or they may also have adverse effect. So what does this tell us? Okay, that medicine is imprecise. So we are treating people with drugs that are ineffective. Why is this happening? Obviously, to put the drug on the market, we need to demonstrate that the drug is effective, and so we make a clinical trial. So the companies identify new molecules, we use them in the mouse model, then we arrive to the human being, okay, and we make a clinical trial. And so let's say, I just take my pointer out. Let's say that this is a patient with some disease, let's say heart problems, okay, and what we do in a clinical trial is that we try to see, so we have two groups, a placebo and a drug one, and we try to see if we benefit, okay, so let's say that this is the output. We will have that some patients have a good outcome, so respond to the treatment, some do not respond, and some have side effects. But the one who drives which drug we will use is the light blue one. Why? Not only because he benefit, but because he was the most abundant in our group. So the current treatment that we used is chosen on the basis of the average patients, so the more abundant. But of course, we also have yellow and red people who do not show in a clinical trial their effect. So this was just a pipeline to show how we usually select a drug, okay? So we have a new molecule, we think it will be good. Let's say that we are in the cancer field. So we take a cancer cell, we culture them in a plate, and then we test the drug, and we say, okay, tumor cells die, so it may be effective. Of course, it is an easy approach, few money, very quick, but cells plated in a plate in 2 dimensions has nothing to do with a cancer who grows three-dimensionally in the body. And so we move to a preclinical model, mice and animals. Of course, animals are complex, so it's not only if it is effective in the tumor, but also if it has side effects, if it reaches the tumor and everything. So very good, but animals are not humans, and so lastly we need to go to humans, but the problem is that humans are all different. Mice or cells are all exactly the same, so of course you obtain data that are clean. When you go to the human, the problem is that you are not allowed to understand which is the light blue, which is the yellow, which is the red. So what is precision medicine saying? Aha, before choosing the drug, let's say before doing a clinical trial, it would benefit the results to understand that we've got at least 3 types of patients. And how can we do this? We can do it by making a precision medicine approach, which says before going to a clinical trial, we need to understand characteristics of the patients, which may be intrinsic characteristics, for example, genomics, so genetic traits of those patients, but this is not enough, and of course it may be lifestyle habits, how they eat, how they fit, and so on. So we merge all this information together, and at the end we will drop out that we've got 3 types of patients. Of course, we will make a clinical trial for the blue, a clinical trial for the yellow, and one for the red, and then we will choose the right drug for those one. Now, just to put it a bit more concrete, I make you an example that historically is the right example. So we start with cancer because precision medicine is born in the cancer field. So we've got a cancer and we say to the patient, you've got a lung cancer, which means that we've done some investigation and we identified that he's got a lung cancer. So at the beginning, we were saying, okay, let's try different chemotherapies to see which will be better. But this was not effective in all our patients. And so scientists said, okay, let's have a look to the patients. So let's start making biopsies to see if by eyes we can realize that there are different types of patients. And for example, in lung cancer, just looking to how the biopsies look and how the cells are arranged, we can identify different types of cancer. So now we don't say you've got a lung cancer, but you can say you've got a spinocellular, an acinar, and so on. Okay, so you identify different type of patients. And you say, okay, now I go to a clinical trial. It improves, but it's not improving that much. So here I identify the different types of cancer. I make an individual clinical trial, but for example, let me show you here that maybe is better. For example, I selected this one, they are all square, so they all look the same histologically, but when I have a look, okay, I've got some that are purple and they will respond, but the other do not. So it's not enough even to go and look what is inside the tumor to say what is going on. What has been the revolution in the precision medicine field? It has been in 1999 when the director of the NIH Institute in the United States said, okay, I will put a lot of money on the table for who will make investigation trying to look molecularly what is going on in the cancer. So, okay, histologically, we identify different types of cancer, but if we look at the mutations that are driving that cancer, can we further improve? And of course, this was the case because we don't say it's a lung cancer, we don't even say it's only a squamous cell carcinoma, but now we say that it's a squamous cell carcinoma with mutation 1, mutation 2, and mutation 3. for whom we identified 3 different drugs. In this way, we refined how we stratify the patients at the very beginning, so doing the clinical trials. So now, for example, our clinical trials are not based only on the histology, but are based on the mutation that have inside. And so what is interesting to see is that with the same mutation, we may find also cancers that look differently, and also cancers that are not only lung cancer, but that behave in other organs, but having the same mutation will respond to the same drug. Why in 1991 he had this fabulous idea to invest in precision medicine? Because the year after, so those were the years where we completed the Human Genome Project. Okay, so there was this big project, 2 big groups in the world, and they were aimed to sequence every nucleotide in our genome. It took 10 years. So for 10 years, 2 big groups were working to do that. But since it was really important, technology started to improve. And today, if I want to make a whole genome sequencing, so meaning I take your blood and I want to know exactly your nucleotides, I will do it in 3-4 hours, okay? So it's not 10 years, it's not even 1 week, it's very, very quick, it costs very lot, and this has been driven by the technological improvement, because now we've got instrumentation that are able to do this. In those years where NIH was funding this research, as you may see, publication increased in this field, demonstrating that the direction was a good direction because we could succeed. And for this reason, in 2015, Obama launched this precision medicine initiative. So he founded with a lot of money a clinic, a cohort study where they collected 1 million people, no matter which race, no matter if disease or not, so volunteers that wanted to participate, and they are starting collecting a lot of data and blood samples in order to follow, because of course the problem of the disease is that you would like to anticipate their occurrence, so you are able to identify predictors, so saying this one will develop this, that one will develop that one. And for this, you need to have them when they're healthy so that you can follow. Of course, genomics, so sequencing of the genome has been one, let's say, of the big step forward. With, let's say, genomics, there is also some other omics related to the same instrumentation. So just to be clear, omics is when we refer to identification of all the molecules of the same type within a sample. So I take a blood sample. If I want to make genomics, I want to know all the genes, so sequence all the genes. If I make proteomics, I want to sequence all the proteins that are in that samples, okay? And so I was saying that together with next generation sequencing, which is the technology to make genomics, there is also mass spectrometry that allowed us to make proteomics, metabolomics, and now the omics are a very big field. What is the problem of omics? So I take a sample and I look for all the molecules of the same type in the sample. What I generate is the so-called big data. I've got a lot of data. I can know all the variations that are within your genome. But now, which is the relevant variation for his disease? I don't know, because I've got a lot of data, and how can I say, okay, this is relevant or not? Just to make you feel the complexity, if we have a look to this picture and I ask you, okay, what do you see in this picture? So what do you see in this picture? Sorry?
Rocks.
Rocks. So he sees rocks. Anyone see something else? Snow. Snow. Rocks, snow.
Mountain.
Sorry?
Mountain.
Mountains. Yeah. So rocks and snow, snow and rocks. Usually when I make it to my students, they say the same. There are rocks and there are snow. Why? Because we are looking at all the data in the same time. But if we would be able by eyes to scan every pixel of this image, what will jump out is that there is a snow leopard lying there and taking sun. But of course, we were not able to distinguish among all these data what was relevant for us. This is exactly what happens with the omics. So now the fields is, we started with genomics, so having a look to all the chromosomes and how they look for, but to understand what is the relevant, we need to put that information in the biology. So for example, if you have a disease and I find 10 variation, to understand which is the relevant one, I need to take that variation to the disease. And so, for example, see if I also find some alteration in the transcript or in the proteins that are encoded by the genes. This is system biology. What does system biology? It takes a lot of information that are not organized and it gives you back a biological information. So this is not just some slices of fruit, but actually when you build it up in a human being, it's fruit salad. So we are moving from this that has been the predictive model till now to a model that is built on precision medicine. So before going to a clinical trial, I make a multi-omic approach. So I study in detail the characteristic, and in this way, I will pop out that I've got the black, the green, they all have lung cancer, but actually they have it in a different configuration, and in this way, if I put them together, I will have a good reply. So is this approach relevant only for cancer, which is the example I made you? No, and the reason why we are here today is exactly this. I just want to show you the building where my laboratory is located in the north of Italy, in Novara, and is the unique center in Italy devoted to study with a multi-omic approach autoimmune diseases. Just to let you show how it is conceived, we've got 2 floors, and in the same building, as it happens for cancer research institutes, we've got the patients, so we've got the clinics, patients with autoimmune or allergic disease. We've got the next generation biobank, which means a place where we can store the samples from the patients, not only the samples, but also the information, what happened that day, which were the drugs, exams, and so on. And then we've got 2 floors. In the second floor, there are our laboratories. And in the first floor, there are companies, so spin-off that are aimed to drive the innovation to the market. My laboratory, so I'm an immunologist, and so I work in the immunology field, and my laboratory is an immunomics lab. Just to take you to my research, so we study the immune system, which is, of course, cells and organs that usually are devoted to fight pathogens, bacteria, viruses, to fight cancer cells, which means our cells that eventually mutated something and start growing without control, okay? And also to distinguish our cells from the one of everyone else, which happens when we have graft rejection. So if I take his kidney, sorry that I take everything from you, but if I take his kidney and I try to put it into my body, my immune system will say, it's cells, it's human cells, but it's not yours, okay? What happens in autoimmune diseases? That the immune system makes a mistake, and so it starts aggressing my cells, not my tumor cells, but my healthy cells, as if they were his kidney, okay? So autoimmune diseases are a broad range of, we've got more than 100, because it depends on which is the cell that is recognized, which is the pathogenetic mechanism, we've got a lot. But what is clear for all of them is that they are mediated by endogenous factors, genomics, but also external factors, how we eat, how we move, and so on. And the project FLAMIN-GO that I'm coordinating, and that is the reason we are here, because of course one of the partners is Science on the Street, and now it is not well animated. It's supposed to pop out here, but anyway, so here in Ljubljana is hosting us. This is one of our meetings. So this project is born in this context, so trying to drive the pathobiology of rheumatoid arthritis, I will tell you in a while what we are talking about, to the precision medicine. So it's called the FLAMIN-GO, which of course recalls of this beautiful bird that you may see has got a joint that is inflamed. But actually, the meaning is FLAMIN-GO, which means we want to make inflammation going away from the joints. Why the joint? Because rheumatoid arthritis is an autoimmune disease, so the immune system aggresses what? Aggresses the joint. And so the joint is the place where two bones are connected and there are movements. So we've got two bones, we've got the cartilage on top of the bone, and then we've got this thin layer, pink layer, which is the synovial membrane, that is very important, a few layers of cell that produce the synovial fluid, which stands here inside, avoiding that the two bones friction one on the other, okay? So they are very important. You may see here the normal situation. What happens in rheumatoid arthritis? Inflammation arrives into the synovia, so the synovia is aggressed by the immune system. The inflammation starts with an acute inflammation at the very beginning, but if we do not control this inflammation, this will evolve over time, causing the aggression of cartilage and bone with damage that will become permanent over time, and these are the consequences of a non-treatment of these patients. Fortunately, this is a situation that is not that common at the moment because we have a lot of drug for rheumatoid arthritis. So over time, we developed different types of drug that are very effective. Meaning very effective, in this case, means that if I give you the right drug, you woke up one morning and you cannot even hold a pen because it was hurting, they were swollen, so it's impossible to make a normal life. But if I give you the right drug, it's like you don't have disease, nothing anymore. You're not cured because you need to take the drug, but you don't have symptoms anymore, and the inflammation do not evolve aggressing the bones. Okay, so what are we talking about today? Why are we here today? The problem is that we don't have any predictive factor able to tell to the rheumatologist which is the right drug for the right patient. So what happens is you come to me and I tell you, okay, you've got rheumatoid arthritis. Usually it takes 1 to 2 years to make a right diagnosis. So in 2 years the disease started to advance. And then I've got the first line. Everyone gets methotrexate, okay? And this is effective for half of patients. So half of patients do not come back, or they just come back to say thank you very much. But the other half will come back after 6 months, because it takes at least 6 months to understand if it is effective or not, saying, okay, nothing changed. And at that time, I've got a lot of drugs, but no way to choose. So what we do is we say, okay, you want to choose? You just pick a ticket at the lottery and see if you win or not, because there's no way to understand which is the right one. And I give you one biologic, okay, and you take it for 6 months, and then you come back, and it's 1 year you started, and nothing happened, and nothing improved. So there are some patients that start cycling from one drug to the other without reaching any benefit, and meanwhile the disease progresses. So, of course, now that you are very friendly with precision medicine, I will say, why don't we put the precision medicine also in the rheumatoid arthritis field, in the autoimmune field? Of course, this is what one of our partners in the FLAMIN-GO, Professor Pizzalis, we've got now Liliane in the audience, started 10 years ago. He started to say, but if in cancer it was so useful to take a biopsy and have a look, why don't I do the same in rheumatoid arthritis? So he developed a way to take a synovial biopsy, it's very easy, and so he started to have a look to the histology, and it jumped out that he was able to find at least 3 different pathotypes. So he found the light blue, the yellow, and the red, just looking at what was happening, okay? But then, of course, the omic era arrived, and so we say, okay, beside looking at what is inside the biopsy, why we don't have a molecular look? It's a mess of figure, but I just want to point out that from 3 different pathotypes, having a look to what is inside molecularly, he improved the number of different patients that were available, exactly as it happened in the cancer field. But now, so it took 10 years to arrive here. And now, how can we select which is the right one for the red, for the yellow, for the green, which is the right drug? We need to go back to a clinical trial. So I need to separate my patients based on the omic analysis and then perform a clinical trial. A clinical trial means a lot of money, a lot of time, and let's say some ethical issue, because when I go to a clinical trial, I divide the patients into the placebo, which means no treatment, and the patients who will have the treatment. So there are several things that say, okay, and this is what I think, the precision medicine approach will be the solution, but it will take a lot of time. So the FLAMIN-GO project was conceived to say, can we make something now that we are not at the precision medicine era yet, to try to find a solution for our patients? So the concept of FLAMIN-GO is completely different. I don't care if you are green, yellow, black, or whatever, because now I'm not able to know. It will be in 20 years. What I want to do instead is to go back to our current approach, and in particular to the cell culture field, which is easy, but starting from cells derived from your biopsies. So I'm taking your cells and I'm trying to model your joint with your cells. Now, of course, having synovial cells flat on a plate is not modeling a joint. What we need to do is to put in the same culture all the cells that are relevant for the joint. We have seen bone, cartilage, the immune system, the synovia, and in the right configuration in a 3D model, so that we are able to model what is going on in the joint. A joint is moving, I want a movement on my culture. What we are doing, so who is answering to this point, is to make an organ on chip, okay? So the idea is I try to model, to rebuild a joint with your cells, so it will be an individual joint of each patient in the world, because I don't care, I don't know which kind you are, I will do your organ on chip. And then I will try to find the right medication. Let's see just 1 second what are organ on chip. So they are microfluidic platform. You see they are very small. What you see here in colors are small channels within which we can put the cells. We can build them with the shape we want, put different cells together, put them on top, put air. We can do what we want. And of course, they are microfluidic because you see there are the medium it flows on top of the cells. So it's not a static situation, but it mimics what happens with the blood. So to build an organ on chip, we need actually 3 things. The first is to design, model, and fabricate the platform, meaning the plate on which we will capture the cells. Then we need to select hydrogels. Hydrogels are a sort of extracellular matrix that allow us to build in a 3-dimensional environment the cells that we want. So we can put them also using bioprinter in the right configuration. And of course, at the end, we need to identify, because to culture 1 cell on top of an organ on chip is very easy. To culture more than 1 cell, it's quite difficult. So to come to our problem, the cells that we want to put in our organ on chip is, of course, the synovia, which is the tissue that is aggressed by the immune system. So, of course, we need to have them there. And since the synovia produces synovial fluid, of course, we need also to have synovial fluid. Then we need to have a blood vessel. Why? Because the inflammation and the aggression of the immune system is taken into the synovial by a blood vessel. And so we need a blood vessel that can make leukocytes, immune cells arriving into the tissue. And of course, we need also to have bone and cartilage, which are not affected at the very beginning. But if I want to model a joint that has rheumatoid arthritis from sometimes, I also need to have those. And so what we wanted to, so this is an enlargement of before, just to show you that we identified a unit, that is the one you may see here, and we try to reconstruct on our chip exactly how the cells are connected one to the other. So we've got a blood vessel with leukocytes inside. These are in contact with the synovial membrane, which is in contact with the synovial fluid, which is in contact with cartilage and then bone. So trying to reconstruct also in a 3D structure how the cells are meshed together. And importantly, since the joint move, we also applied in the synovial fluid pressure, allowing us to put pressure and remove pressure, simulating the movement. Okay. So let me just give you the concept at one glance. We can start from no matter which color of patients, because we don't care. What I do is I take synovial biopsies. Starting from the cells that I find inside there, I will obtain all the cells that are different one from each other, and then bioprint them into this small platform in the right configuration. Starting from one biopsies allow me to have enough cells to make 40 different synovial platform at the same time, which means that I tell you today you've got rheumatoid arthritis, I take you one biopsies, and after 1 month I will have 40 replicates of your joint, and I can test all the drugs that are available on the market, identify in few hours which is the right one for you. So this is a consortium coming from different parts of Europe that is involved in this project. Each of them have a different part. I think that you understood it is very complex what we are trying to do. We are almost at the half time of our project, so we were able to build individual tissue, so a blood vessel on a chip, a joint, osteochondral unit on a chip, a synovial unit on a chip, and now we are trying to merge all these data together. Nevertheless, the results are very encouraging, and so we started by making a company, a spin-off of two of our partners. So one is UPO, which is us, and the other is CNR in Italy, trying to take into the market a part of the results that are on this, especially related with the synovial part. So the business model of our company is, so what I aimed is to take, I think in 10 years, it will not be tomorrow, but I will take into the hospital the platform, meaning that the patients, when they receive the diagnosis, they immediately can give their biopsies and have very quickly their reply. This is what I wanted to tell you, and thank you very much for your attention. And I had a last slide, but I don't see it now. So, see, it is this one just to show you my group at CAD and also the founding, not only the U1, but also the other. Thank you very much. It's a very relevant question. Actually, I didn't tell. The platform are transparent, so they can be put under no matter which microscope. You can stain the cells if you want to, since there are different types of cells. We put immune cells which are different, so you can stain them to see in fluorescence, for example, to see if they behave or not. You can see if they live or die, for example, using a time lapse, so following over time. Of course, yes. It's not really a matter, at least the one we conceived, of penetration, because the amount of tissue that is inside is very thin. But in any case, it is possible to do, yeah.
So does something like this, I think I've heard about it, already exists something like this also for the cancer, because for the cancer it's very difficult, to reach the core of the cancer.
Yeah. Actually, there are different architectures and types of organ on chips you can do. For example, in cancer, especially some cancer, you make organoids, which means that it's not cells really put into a chip, but it's more rounded shape, so you can really understand what goes inside and what not. There was also, yeah, a question. Okay. Yeah. It depends, obviously, on which kind of tissue we are talking about. Now, staying on synovial cells, synovial fibroblasts, it has been already very well, so Liliane worked a lot on this, but it has been described in the literature, that when you take the biopsies, of course, you've got a lot of cells inside, different types of cells. Each of them will last for a while, but if you split the cells 3 times, you will get the great majority of synovial fibroblasts, which are still, let's say, a bit, So, starting from the 3rd passage, we can decide to hold them as a synovial fibroblast, and so keep on until passage 7. After passage 7, they lose their capability of build a real synovial membrane. But in the 3rd passage, if we add some factors that may push differentiation to chondrocyte, to osteoblast, we still have inside cells able to give those. And this is the reason why we start not making a biopsy of the bone, but the same biopsy will give the cells to build all the tissue. Yeah?
So when you have cells in the flask, you change medium, they grow, they can die, you can see are they stuck to the bottom or they're not. How do you see that in the chip? And also, is there different medium for different cells? Or is it the same one? How do you actually change it?
Okay, so the medium, let's make a basic organ on chip with one cell type, okay? So we build the chip and then we start the microfluidics. It means there is a pump that pushes the medium inside, it stays and then it goes and then you can make it circulate, you can add drugs or something, okay? At the very beginning, when we start the characterization, we make models to see if the medium was arriving in every part of the chip, and this is the case. So we set the pump in a way that the nutrients arrive all over. Okay. You can also retrieve what is coming out and, for example, see if it is inflamed, not inflamed, if there are some metabolites that comes out. We are studying also extracellular vesicles, so we collect also those. And of course, you can have a look going through the zeta stack of the culture, how the cells behave over time. This is what no. But yeah, the hypothesis is that it is. No, that is the end point of our FLAMIN-GO project, because Professor Pizzalis already has a lot of clinical trial ongoing where he takes the biopsies, so we have the cells, but he also knows which patient is responding to which drug. So at the end, when everything will be fine, we will make a pilot clinical trial with the patients we already know how they respond. And at that time, we will come back in Ljubljana and we will say, give you the response. Yeah, this will give us an idea if we need to go on this or not. But of course, to really respond to that question, we will need to make a multicentric clinical trial where we collect the samples from different parts to see if really having good big numbers, because here we will have, 10 patients, which is something, but of course it's not enough to really say that this will be the future for rheumatoid arthritis. But just to make a last, there are a lot of autoimmune diseases that are in the same condition, so they've got no cure, or the cure we do not know how to allocate, and that this is not the case of rheumatoid arthritis, but many others are currently biopsied. So it's in the clinical practice to biopsy them. And so, for example, salivary gland, there are lupus nephritis, there are a lot. So ideally, the concept can be spread in different conditions with what we already do in the clinics.
Okay, thank you very much. Thank you very much for your kidney.
Yeah, it was.
Thank you. Let's thank you.
I'm sorry for being late. Are we staying here?